Good evening, everyone. Let's begin with today's session. And as we say, let's crack NEET PG together. Let me just check whether I'm live or not. Let me just check what's the issue. Okay. So, fine. Let's begin with today's session and let's crack NEET together. So, today we are going to discuss a very interesting topic, that is your tryptophan and glycine metabolism. So first of all, we'll be discussing tryptophan metabolism and then glycine metabolism. But before beginning with the actual topics, let us have a small introduction about Unacademy. So Unacademy is one of the India's largest platform for the preparation of NEET examination. And you can get the best educators all over India on this platform. And what is so special about Unacademy is that you can attend these special classes, and in these special classes, you will be getting one-to-one interaction with faculty members. They'll be easily solving your doubts, and they are always available for any of your query and doubts. Then there is a test series, and which is very much important because for any examination, you know how much you have learned so far. So with the help of test series, you can easily evaluate how much you have learned so far. So this is also possible with Unacademy, that you can easily go through the test series. Then it is very easy to start learning. You can just go to Play Store and write Unacademy, and you will see the logo of Unacademy Learning app. And when you will see the logo of an Academy Learning app, what you need to do is you need to just download the app and make your account, and you are good to go. So it is very easy to start learning. Then what are the other features with online learning is there is flexibility of schedule. You can learn anytime, anywhere. It is very much easy. Then language can be of your choice, like English, Hindi, and various other regional languages are also available. Many faculty members, they try to make the things easier for the students, and they teach in the regional languages. Then unlimited practice. Once the quiz has been taken and a video has been recorded, then you can easily go through the videos, and you can always take the quizzes as many number of times as you want to. So unlimited practice is one of the another benefit with Unacademy, right? So what you can do is you can just go to the app and you can write the Unacademy term, and you will be directed towards the course, and then you will need to choose the course over there, and you can start your learning, right? Good evening, Porus. Good evening, Jeevan Rathore. Then it is almost classroom like feeling. You won't feel anything different from a normal classroom. You can chat with your educator, then there are interactive polls, and with the help of interactive polls, what you can do is you can just answer the questions which your faculty member is asking you, and you can also come to know that the students, they are having doubt in which options, like which options are even overlapping for other students as well. So interactive polls are very helpful. Then your teacher will be explaining each and every option. So interactive poll is quite interesting as well as quite uh, informative in terms of learning. Then you can learn anytime, anywhere, because once the video is recorded, then there is no bar of the timing at what time you are learning. And obviously, anywhere, if it's a live video also, then you can attend it from anywhere you are, right? Then there is another benefit with Unacademy is it provides you with Plus platform. And what is so good about Plus platform is you will be getting all the 19 subjects videos sequentially. Each and every topic will be covered on this Plus platform. And for any of the upcoming exams, faculties, they keep on conducting the short-term courses and crash courses. So you can anytime go and attend these courses. So Plus platform will be highly beneficial in any kind of preparation. So this Plus platform subscription is for 1 month, 3 months, 6 months, 12 months, and 24 months. And what is my personal suggestion is go for a 1 yearly or 2 yearly subscription, because then whatever videos are available over there, you can make the most out of the content over there. So go for a yearly or a 2 yearly program and prepare well for the examination, and definitely you will qualify it. So the amount that you need to pay for this Plus subscription is quite less. It is quite pocket friendly. Okay, and in that amount also you can get an additional 10% discount if you are using my referral code, that is FB01. So just use my referral code FB01 and avail an additional 10% discount, right? So let's start with today's class, that is tryptophan metabolism and glycine metabolism. So first of all, let us understand what is tryptophan metabolism. So these amino acids, they are divided into various categories, or they are classified into various groups. So one of the classification of amino acids is on the basis of its requirement, whether the body can form it, or whether the body cannot form it, or there are some amino acids which are partially formed in the body and partial amount is needed from outside the body. So essential amino acids are those which you need from outside the body. So tryptophan, the one which we are going to today's class, belongs to the category of essential amino acid. You need it from outside the body. Then conditionally essential amino acids, like they are semi-essential in nature. Some of the amount can be formed in the body and some of the amount you need from your diet. The non-essential amino acids are those which can be easily made in the body. So your glycine metabolism, the second topic which we'll be discussing today, falls in the category of non-essential amino acid, right? So tryptophan is your essential and glycine is your non-essential amino acid, and it can be formed in the body, right? Then they are also divided on the basis of their R group. Like this amino acid, it has got the basic COOH and NH2 and H and one of the R group. So this R is variable in all the amino acids. So depending on this R group, the amino acids are classified into various categories. So your tryptophan, it belongs to the category of aromatic amino acid. See over here, it has got two rings and it belongs to the category of aromatic amino acid. Then second, we are going to see glycine. So glycine is a very simple amino acid. So in place of R, we need to put only H. So glycine belongs to the category of non-polar aliphatic R group containing amino acid. So it's non-polar aliphatic. And tryptophan is aromatic and aromatic R group containing amino acids, right? Then talking about exclusively about tryptophan, let's start with tryptophan metabolism. So tryptophan, we have seen that it's an aromatic amino acid. Already we have seen that it's an essential amino acid, and we have seen that it is made up of two rings. So this two rings is called as indole group. The two rings in tryptophan is called as indole ring. See over here. One ring is containing 6 carbon atoms, and the another ring, it contains one NH, and this total ring is called as indole ring. So special group in tryptophan is indole group. Then it is a kind of glucogenic and ketogenic amino acid. There are amino acids which belong to these categories. Some are glucogenic and some are ketogenic. So amino acids which belong to ketogenic and glucogenic acid, or we can say the category of both glucogenic and ketogenic amino acid. Examples are phenylalanine, isoleucine, tyrosine, and tryptophan. So these four amino acids are the one which belong to the glucogenic as well as ketogenic group, right? So your tryptophan is not alone in the category of glucogenic or ketogenic amino acid. There are three friends of tryptophan, that is phenylalanine, isoleucine, and tyrosine, and it has got a indole ring, right? This is the basic on the basis of structure and everything. And now let's move further. Now, when we are saying that we need to break down tryptophan, so the substances which are formed during the breakdown of tryptophan, they are alanine, and alanine will further lead into the glucogenic pathway. That is, it will enter into your glycolysis. Then if it is we are saying ketogenic, it means it will further form fatty acid. So the fatty acid or the ketogenic pathway is further led by acetoacetyl CoA. Then formyl group, one carbon unit is formed from tryptophan. Niacin and NAD is formed from tryptophan. Serotonin is formed with the help of tryptophan. Melatonin is formed with the help of tryptophan. And HII, hydroxyindole acetic acid, that's an excretory product. It is also formed with the help of tryptophan, or can say with the breakdown of tryptophan. Right? So these are the 7 substances which are formed from tryptophan. We will be taking each and every one individually. So this is a kind of table which is showing you the summary that which are the intermediates while the formation of the products which we are saying. So tryptophan, it will formation of one carbon unit formyl group and formyl kynurenine. Then serotonin and melatonin is formed. Then alanine will enter into glucogenic pathway. Then acetoacetate will further enter into ketogenic pathway, and niacin and NAD form from the same kynurenine pathway. So each pathways will be dealing individually. Now let us talk about the metabolism. So when we are saying that tryptophan is an aromatic, we have discussed it's an aromatic, essential, and indole ring containing amino acid. So its major fate, the first reaction which will occur in tryptophan metabolism is that the tryptophan will undergo is to be oxidized or oxidation with the help of tryptophan pyrolase. Tryptophan pyrolase will first of all act on it, and it is a hemoprotein, that is it contains, it needs iron, or it is an iron containing, let me take a pen. Yeah, so it's an iron containing enzyme, the tryptophan pyrolase, and the enzyme is inducible by corticosteroids. And when this reaction is taking place, we'll be the table also, we'll be just going through the table also, just I'm the outline and then we'll move to the table. So this reaction, the first reaction when tryptophan is acted upon by tryptophan pyrolase, that is an iron containing enzyme. What happens is one of the oxygen molecule will enter into the pathway. In this major pathway, total 11 carbon atoms of tryptophan are catabolized into formyl group. So total 11 carbons of the tryptophan will be broken down. In the major pathway, kineurinase is an enzyme dependent on pyridoxal phosphate. So when this happens, when your tryptophan is acted upon by tryptophan pyrolase, what happens is N-formyl kineurin is formed. Let me write over here. N-formyl kineurin is formed. And when this kineurin is formed, what happens is there is another enzyme which will form kineurin from this, and urinin will be further acted upon. So therefore in vitamin B6 deficiency, the pathway will be blocked at this level and it leads to the niacin deficiency and manifestation of the pellagra-like symptoms. So the accumulated kineurin is shunted to xanthurenic acid. So in pyridoxal deficiency, xanthurenic acid is included in urine. So let us just see the pathway. This is tryptophan, and this tryptophan, there is an oxygen molecule which is required in this very first step, and the enzyme which is required over here is tryptophan pyrolase. Tryptophan pyrolase, and this is the enzyme which is iron containing, right? And when this enzyme acts upon tryptophan, it forms N-formyl kineurin, right? And when this N-formyl kineurin is formed, there will be removal of one formyl group one from this, and this is assisted by vitamin B2, assisted by vitamin 2. And the enzyme which is actually responsible for removal of one of the formyl group is formylase, right? So there will be removal of one of the formyl group, and the process is B2 vitamin B2 assisted, and from then what is formed is kineurin. Kineurin is formed. Now this kineurin is converted into 3-hydroxykineurin, and the enzyme which is responsible over this at this stage is kineurin hydroxylase. So for kineurin, I am writing K and hydroxylase. Hydroxylase. And this kineurin is converted into 3-hydroxykineurin. Right? 3-hydroxykineurin. And if there is some defect in the further pathway, it will be directed towards the formation of xanthurenic acid, and then this xanthurenic acid will be excreted out in urine, and it is manifested as pellagra-like symptoms, and there will be deficiency of vitamin B6. There will be deficiency of vitamin B6, and this will be excreted out in urine, right? So this is till here. Now if 3-hydroxykineurin is formed, if it is formed, then what will happen? If it is formed, then it will further, oh, and over here also there is one thing, 3-hydroxykineurin, and one of the thing that is formed from the formation of 3-hydroxykineurin is alanine. And from here, the glucogenic pathway of tryptophan is diverted. This alanine formation from 3-hydroxykineurin diverts the pathway towards glucogenic pathway. Then after the formation of 3-hydroxyanthranilic acid, it will be further forming amino carboxy mucoaldehyde, or it will be diverted towards the NAD pathway. Right. So first of all, we are dealing with the formation of completion of this pathway. So what will happen that from the formation of amino carboxy mucoaldehyde, there will be removal of carbon, one of the carbon dioxide, and amino muconate aldehyde will be formed, and which will further lead into formation of acetoaldehyde, and this acetoaldehyde will be further directed into ketogenic pathway, and this is how the ketogenic pathway occurs, and glucogenic pathway occurs in tryptophan metabolism. So some of the drinks structures are also given over here for a better understanding. But what you should remember, at which stage it is entering into glucogenic pathway and at which stage it is entering into ketogenic pathway, right? So we have understood that well. Now the second thing that happens with the tryptophan metabolism is the formation of niacin or nicotinic acid pathway. So we will be starting with nicotinic acid pathway. So what happens in this pathway is about 97% molecules of tryptophan are metabolized in the major pathways. About only 3% of the molecules are diverted into hydroxyanthranilic acid form NAD. Like the major pathways which occur in tryptophan metabolism is your glucogenic pathway and ketogenic pathway, and rest all of the most of the thing will go over there, and only 3% will further form this NAD. So the enzyme that is responsible, the primary enzyme that is responsible is QPRT, that is quinolinate phosphoribosyltransferase, transferase, and it's a kind of rate limiting step, that is it is the lowest step of the process. About 60 milligram of tryptophan will form just 1 milligram of nicotinic acid, and that is also very much important. So development of pellagra-like symptoms in maize eating population is due to tryptophan deficiency in maize. Hello lionfish. Hydroxyanthranilate production is dependent on pyridoxal phosphate, and hence in vitamin B6 deficiency, nicotinamide deficiency is also manifested, right? So let us look at the actual process of this formation of NAD. See over here, there is this tryptophan, and this tryptophan is getting converted into N-formyl kynurenine, and we have understood kynurenine formation, 3-hydroxykynurenine formation, and at the place of 3-hydroxyanthranilic acid, this is the place where the process will be diverted. So 3-hydroxyanthranilic acid will further form phenolinic acid and NAD. So how does this happen actually? So when we were seeing the glucogenic and ketogenic pathway, there we also saw that there will be formation of amino 3-carboxy muconate succi semi aldehyde. When this is formed, it can lead to three fates. There will be three fates, and the enzyme which will be acted upon this will be picolinate carboxylase. Picolinate carboxylase. Picolinate carboxylase. See, so this is the enzyme, and when this enzyme will act upon, it will lead to formation of picolinate. Those who are attending the class over here now, please write in the comment box whether my voice is clear or not, because some of the students, they are facing the problem of unclear voice. So please comment, write in the comment. So one of the fate will be picolinate formation. There will be removal of carbon dioxide. Then there will be formation of phenolynate, and there will be formation of acetyl CoA. Right? This will be formed, and acetyl CoA will further enter into formation of fat, and this quinolinate will form nicotinamide mononucleotide, and which will further form NAD and NADP positive. So this is how the process occurs. So one of the most important enzyme which you need to remember, which we discussed in the written format also, that was your QPRT. Let me write over here, QPRT. And what is the full form of this QPRT? Quinolinate phosphoribosyl transferase C. Quinolinic acid phosphoribosyl transferase. This is the enzyme which is responsible for formation of NAD and NADP. So we have seen how your glucogenic pathway occurs, how your ketogenic pathway occurs, and then how the formation of NAD and niacin occurs with the help of tryptophan metabolism. So here, this is in the form of the structures. You can see the form of the structures. This 3-hydroxyanthranilic acid will form quinolinic acid. There will be just shuffling of some of the things, and there will be removal of carbon dioxide. There will be formation of niacin or nicotinic acid. So you can see the structure over here, how the niacin or nicotinic acid it looks like. And then this process is energy dependent conversion of niacin to nicotinate mononucleotide. See, one molecule of pyrophosphate is released over here, and then this process is again energy dependent, and it leads to the formation of amido NAD, and which will further, with the help of energy dependent reaction, gets converted into NAD positive, right? So this is how your NAD positive is formed with the help of tryptophan metabolism. Then another important thing that is formed in the pathway is serotonin formation and melatonin formation. So both of the pathways are quite linked, and we'll be seeing then that how serotonin and melatonin is formed. So it can be compared, or it can be just thought in a way like how the synthesis of epinephrine and norepinephrine occurs. It's similar to epinephrine and norepinephrine formation, right? So what happens is serotonin, what its actual name is 5-hydroxy tryptamine. So remember its word 5-hydroxytryptamine, because there are PYQs which are based on the name of serotonin. Just they'll be asking the name of serotonin. So serotonin is produced in, like, where it is produced? It is produced in brain cells. It is produced in mast cells. It is produced in platelets. It is produced in gastrointestinal tract mucosa. These are the places where your serotonin is formed. So tryptophan is first hydroxylated by tryptophan hydroxylase, and this reaction is very much similar to phenylalanine hydroxylase. The coenzyme which is needed over here is tetrahydrobiopterin. The tetrahydrobiopterin is regenerated using NAD, and it is very fastly regenerated. The coenzyme that is needed is tetrahydrobiopterin, right? And in the next step, 5-hydroxytryptophan is decarboxylated into 5-hydroxytryptamine. So it's not a very long metabolic pathway, it's a quite short one. And just in 3-4 steps, it will be completed. There will be formation of serotonin, and there will be formation of melatonin, both of the things. The enzyme required over here is uh, decarboxylase, right? So just have a look over here. This is the actual mechanism. This is our tryptophan. This is our tryptophan over here, and it is a coenzyme dependent reaction. So the coenzyme that is required is tetrahydrobiopterin, and we just read that it is regenerated with the help of NADP. So it is regenerated, and how this tetrahydrobiopterin, it was converted into dihydrobiopterin, and it got again regenerated there only. And in this process, what enzyme is needed? So let me take a pen and we'll write the name of the enzyme also over here. The enzyme needed over here, pen. So the enzyme over here is tryptophan hydroxylase. Tryptophan hydroxylase. The enzyme needed over here is this. Then when 5-hydroxytryptophan is formed, then there will be a decarboxylation reaction. Can you see this carbon molecule going out? So this is a kind of decarboxylation reaction, and in this decarboxylation reaction, just in 2 steps, your serotonin is ready. So serotonin is formed. 5-hydroxytryptamine or serotonin is formed, right? And as I said that the formation of melatonin and serotonin is just linked. So just taking the process ahead, there will be an expenditure of acetyl-CoA, and there will be addition of acetyl group to in the serotonin, and it will form acetyl serotonin. So when acetyl serotonin is formed with the help of acetyl-CoA, the enzyme that is responsible for this kind of reaction is acetylase. So let's write the name of the enzyme over here, acetylase. Acetylase. This is the enzyme which is participating in the process. Now, this is your 5-hydroxytryptamine. It can also form further HIAA, that is hydroxyindole acetic acid. Right? This hydroxyindole acetic acid is a kind of excretory product, and it is excreted out in urine. So this is also linked. So when we are talking about formation of serotonin, and we are talking about formation of melatonin, the formation of HIAA is also linked in the process, right? So acetyl serotonin is formed over here, and with the help of our universal ethyl group donor, that is our SAM, our melatonin is formed. So what is the name of melatonin? Like the name of serotonin was 5-hydroxytryptamine. So we need to know about the name of melatonin also. Yes, we'll be Jeevan Rathod. Yes, we'll be discussing the three type of diseases related with the tryptophan metabolism, right? So the name of melatonin is N-acetyl S-ethoxyserotonin, right? And what is formed? When SAM was ex expended at this reaction, what is formed? SAH is formed, and the enzyme responsible at this particular stage is methyl transferase. The enzyme responsible is methyl transferase. So this is how the process of formation of serotonin and melatonin occurs. Remember the name of serotonin, that is 5-hydroxytryptamine, and there is a coenzyme which is responsible for this process is tetrahydrobiopterin, and this is how the process of serotonin and melatonin formation occurs. Now let us have a look about the functions of serotonin. Let me read later. Yeah. So what are the function of serotonin? It's an important neurotransmitter in brain, and in brain cells, serotonin is in the bound form. The neurotransmitter found in brain, then it is in the bound form. Then 5-hydroxytryptamine is an antidepressant as well. So the serotonin is kind of an antidepressant as well. When ordinary proteins are taken, all amino acids are available in blood. This causes traffic jam in the amino acid transport system in the brain cells. Tryptophan, the bulkiest amino acid, is therefore taken up very slowly. Tryptophan has got this indole ring, that's why it is the bulkiest amino acid, and being so bulky, it is taken up or it is absorbed very slowly in blood, right? So let us move towards more function. However, when carbohydrate-rich diet is taken, insulin secretion is increased, which will lower the amino acid concentration in blood. So tryptophan easily enters the brain cells. When tryptophan is available in brain in excess quantity, serotonin may be generated, and this is how it induces sleep. So this how it induces sleep with the help of the melatonin, how the formation of melatonin will occur, that is indirectly related to how tryptophan is formed, because your melatonin is responsible for sleep and wake cycle. So this is how it will induce sleep. Insulin secretion will increase, and which will lower the amino acid concentration in the blood, and thus tryptophan will be available, or it will easily enter the brain cells, and when it will be in large quantity, will induce sleep. So carbohydrate will induce sleep, while protein-rich diet will cause alertness and sleep. And if they are in proportionate quantity, then if your carbohydrate is in large quantity, it will just lower down the quantity, and it can induce sleep. Serotonin is found to be low in patients with depressive psychosis. So in depressive psychosis, the serotonin level is low. Serotonin is involved in mood, sleep, appetite, and temperature regulation. These are the functions of serotonin. It increases gastrointestinal mobility as well. Increases gastrointestinal mobility. Sensitivity to pain is reduced by serotonin. Serotonin also reduces sensitivity towards pain. So this is how the functions of serotonin are, that it is responsible in pain perceiving mechanism, then it is involved in mood, sleep, and appetite and temperature regulation as well. Now, catabolism of serotonin. So we have just seen the catabolism of serotonin, that how it will enter into formation of melatonin. So let us see individually that MAO, that is monoamine oxidase, converts serotonin to 5-hydroxy acetic acid, that we have seen also in last slide, that how it is formed. So formation of HIAA will occur, and MAO will oxidatively aminate the compounds having an amino group attached to the carbon atom, and this is similar to the degradation of epinephrine. So I told you in the starting of serotonin that you can relate it to the neurotransmitter uh, metabolism, that is epinephrine and norepinephrine. So the degradation is quite similar to epinephrine. MAO inhibitors will cause mood elevation. So if there will be less catabolism of serotonin, then serotonin will remain in the active form for a long time. And so MAO inhibitors or monoamine oxidase inhibitors are usually used for the depression symptoms. So the drug that is given is iproniazid. Small portion of serotonin is conjugated with sulfate or with glucuronic acids, and they are excreted out in the urine. So small portion of serotonin, they can conjugate sulfate compounds or with glucuronic acid, and they can be also excreted out in the urine, right? Then there was a student, he was asking about the diseases. So yes, the disorders we are going to deal now. The three disorders which are related to fan metabolism, that is Hartnup disorder. The first one is Hartnup's disorder. So from where this Hartnup's name came, that this name belongs to the person who was infected, who got this disorder. So first family member in which the disease was described. So from there the word is coming from. So it's a kind of inherited autosomal recessive disease. The kind of inherited recessive disease. Then absorption of aromatic amino acids from intestine as well as resorption from the renal tubules are defective. So amino acids are excreted. So amino acids are excreted in urine. So amino acids are excreted in urine, right? So there is defect in the absorption of tryptophan from intestine and renal tubule, and 60 milligram of tryptophan leads to formation of 1 milligram of niacin, and if the mechanism is defective, it will also lead to pellagra-like symptoms due to deficiency of niacin derived from the tryptophan, and the manifestation of the problem is dermatitis and ataxia, right? So let's move to the next disorder, that is carcinoid tumor, or it is also called as, let me, it is called as carcinoid tumor, or it is also called as argentafinoma. Argentafinoma. So what happens is the serotonin, it is formed in brain cells, mast cells, platelet cells, and intestinal cells. The formation of serotonin in intestinal cells is with the help of argentaphin cells. So whenever there will be a tumor of argentaphin cells, 60% of tryptophan will lead into the formation of serotonin, and this is how the carcinoid tumor occurs, right? So what we said, that formation of serotonin occurs normally in brain, mast cells, platelets, and intestine. So formation of serotonin in intestine occurs in the argentaphin cells, and when there will be a tumor of argentaphin cells, it will lead to 60% diversion of tryptophan into formation of serotonin, and thus carcinoid tumor occurs, right? Then comes is xanthurinate in urine. So what happens in B6 deficiency, excretion of xanthurinate in urine occurs. If B6 is deficient, then what will happen? This xanthurinate will be excreted out in the urine, right? So tryptophan metabolism is dependent on B6 status because kynurenine requires PLP, and this enzyme is very sensitive towards B6 deficiency. So tryptophan load test is used to establish B status, determination of excretion of kynurenine, and xanthurenic acid indicates the what is the B6 nutritional status is, right? So then I have included one or two MCQs. There are some students presently in the class, so I would like if they can just answer these MCQs. So I've included just two, three. So clinical symptoms of heart nerves disorder include, yes, there are some of the students who are live in the class. I would like them to answer the question. Yeah, anyone? I'm waiting. Yeah, is there anyone? D, all of the above. Jeevan Rathod and lionfish. Okay, great. Then amount of tryptophan that is converted into serotonin. Amount of tryptophan that is converted into serotonin. Amount of serotonin that is amount of tryptophan that is converted into serotonin. No one? No one is answering this? That how much amount of tryptophan will be converted into serotonin? Okay. Answer is 4%. Amount of tryptophan that is converted into serotonin. The answer is 4%. Right? So let us move further in glycine metabolism. Fine. So we have completed our tryptophan metabolism, and now we are moving towards the next part of our class, that is glycine metabolism. Okay, glycine metabolism. So coming to glycine metabolism. So we have seen about glycine, the basic things we have seen since starting of the class, that glycine is the simplest amino acid because the R group that is present in glycine is as simple as your H. Instead of R in other amino acids, R group is quite complex, but when we talk about glycine, it is a simple H. The non-essential amino acid, and it is the glucogenic amino acid, right? It is non-essential and it is glucogenic. Then from where this glycine is formed, let us read about that. First of all, it is formed from serine, it is formed from threonine, it is from the glycine synthase, formed from glycine aminotransferase, and we'll be starting with how it is formed from these things. So from serine, the beta carbon of serine is channeled into one carbon pool carried by tetrahydrofolic acid. Alpha carbon of serine becomes the alpha carbon of glycine. We'll be just seeing the structure also. So the beta carbon of serine is channeled into one carbon pool and carried out by tetrahydrofolic acid. Alpha carbon of serine becomes the alpha carbon of glycine. So another thing, it is also formed from threonine and the activity by threonine aldolase. Right? Let us see how. See over here in the first picture, this you can see. So this serine in the presence of THFA, it is converted into methylene THFA. Its beta carbon will go over there and its alpha carbon will, as it is, enter into glycine, or it will just give rise to glycine. Fine. Then the second thing that we are going to talk is formation of glycine from threonine. This is the structure of threonine in the presence of enzyme threonine aldolase. What happens is your glycine is formed and one of the acetaldehyde is formed. So two carbons of your threonine goes in the formation of acetaldehyde, and the other carbons, they remain in the glycine. So the alpha carbon remains in the glycine and rest of the carbons, they go in the acetaldehyde. Right? Now from glycine synthase. Glycine can be synthesized by glycine is also, and reaction from CO2 NH3, one of the carbon units. And this is the reversal of glycine system. Glycine is broken down, it can just reverse back in the reaction. It's a multi-enzyme complex, needs coenzyme NAD, lipomine, tetrahydrofolic acid, and pyridoxal phosphate. All is needed in glycine. This is from glycine synthase, and it's a kind of de novo synthesis of glycine. Then glycine amino transferase can catalyze the synthesis of glycine from glyoxylate and glutamate or alanine. So glycine aminotransferase, what it will do, that it will form glycine from what all things? Glyoxylate, glutamate or alanine, and this reaction strongly favors the synthesis of glycine, and glycine formation will be highly favored. So you can see over here your glycine, and glycine is converting into amino methyl group and further into NH3. So this reaction is reversible. Reaction can be reversed back, and thus the glycine formation can occur from the formation of its breakdown products as well. Fine. Glycine is completely degraded to CO2, ammonia, and one carbon unit, that is DHFA. The reactions are readily reversible when the when the enzymes are together, called glycine synthase. So over here the same thing is being explained, its degradation, and the reaction can go in the another direction also. So that it, what can be said, that it is a kind of reversible reaction. It's a kind of reversible reaction. Glycine, glycine is forming amino methyl group, and then it is breaking down, and this is how your glycine is broken down. But with the help of glycine synthase, the whole reaction can be reversed back. That is the thing. It's a kind of reversible reaction, and when it becomes reversible, when there is a presence of enzyme called as lysine, so the enzyme will lead to formation. Right? Let's further utilization of glycine, where it is utilized. The first thing, glycine cleavage system. Glycine cleavage system. So glycine, it undergoes oxidative deamination from NH3 and CH2 and one carbon unit, that is THFA. This we have seen just now in the last slide. Pathway is a major catabolic route for the glycine. This is the major catabolic product or metabolic pathway of glycine breakdown. Then the enzymes which are responsible is glycine decarboxylase, and it requires pyridoxal phosphate and lipoamide containing amino methyl transferase. These are the enzymes which are responsible in glycine cleavage system. Then methylene DHFA synthesizing enzyme and NAD dependent lipoamide dehydrogenase. This enzyme is anchored to an H protein. Then another thing that we are saying, that it's a glucogenic amino acid, and tryptophan was our glucogenic as well as ketogenic amino acid. Here it is a glucogenic. So how is it entering glucogenic pathway? Glycine is mainly channeled into glucogenic pathway by first getting converted to serine. It will be first converted into serine and then it will enter into glucogenic pathway. This is the reversal of serine hydroxyl methyl transferase reaction. The reversal of serine hydroxyl methyl transferase reaction, just a reversal. Is this then converted into pyruvate by an enzyme that is serine hydratase? Is then converted into pyruvate with help of serine dehydratase, and then with help of pyruvate it will be entering into the glucogenic pathway. So here I have attached the reaction forming lysine. So and then this serine, when lysine will go back into formation of serine, it can form amino acid, and this amino acid will further enter into glucogenic pathway, that is the formation of pyruvate, and how the reaction will occur. Here the reaction is this direction. Glycine will formation of serine, and serine eventually into formation of pyruvate. The special metabolic functions of glycine. What are the special functions of glycine, or what all things are formed from glycine? So creatine phosphate, and creatine. This creatinine, that is another thing that is produced from glycine. Then heme nucleotides are formed from glycine. Glutathione is formed. Conjugating agents are formed, and neurotransmitters are formed from the glycine metabolism. So in this picture, there is a complete list of all the things that are formed from the glycine metabolism. Proteins and threonine, they will lead to formation of glycine. And when this glycine will be broken down, or glycine can also be formed from de novo sources, that is carbon dioxide, ammonia, and one carbon unit. So these four things, protein, serine, threonine, and de novo, these all things will lead to formation of glycine. And when glycine needs to be broken down, it what will happen? It can go into protein, like CH system. It can form urea or one carbon unit. It can form serine, which can further enter into pyruvate or glucogenic pathway. It can form cysteine or choline. Then glycine can lead to formation of oxalate. It can lead to formation of creatine, creatine phosphate, and finally creatinine. Or it can give the things in purine ring, that is the 4, 5, and 7 positions of the purine ring, or it can form heme, or it can form glutathione, or it can form bile salts, or it can form uric acid through conjugation pathways. So this is how the glycine will further be broken down. So creatine and creatine phosphate. The word creatine is derived from Greek term, that is creas or flesh. So creatine constitutes about 0.5% of total muscle mass, and it is synthesized from three amino acids, that is glycine, arginine, and methionine are the amino acids which participate in formation of creatine and creatine phosphate. So here this is the picture that how creatine and creatine phosphate and finally creatinine is formed. See, glycine glycine will lead to formation of guanidoacetic acid, and in guanidoacetic acid there will be a transfer of methyl group, and when methyl group is transferred it will be formation of creatine, and with the help of energy expenditure process it will contain one of the phosphate group and it will get converted into creatine phosphate, and with the expenditure of one of the inorganic phosphate, what will be formed? Creatinine will be formed. This is how the process of formation of creatinine occurs. And here, as you can see, this is a kind of coupled reaction. The kind of coupled reaction. So in this coupled reaction, what is happening is arginine is getting converted into ornithine. So instead of mentioning ornithine, we can just write glycine will react with arginine and it will lead it into formation of creatine and then creatine phosphate and then creatinine. So this is a SAM dependent process because it's healing group. This is how the formation of creatinine occurs. And what are the clinical application of this thing? The normal creatinine level is zero point 0.7 to 1.4 milligram per deciliter, and serum creatine level is zero point 0.2 to zero point 0.4 milligram per deciliter. Creatine level in blood is a sensitive indicator of renal function. We know it very well. Urine contains negligible amount of creatine in normal males. If it's normal male, then there will be no amount of creatine seen in the urine. So we know this very well. Then in muscular dystrophies, the blood creatine and urinary creatine level is increased. The level of blood creatine and urinary creatinine will increase. Enzyme creatine kinase is elevated in myocardial infarction. So in MI, it is a earliest early indicator, right? Because as soon as heart attack occurs or as soon as MI occurs, the level of creatine increases very rapidly. So it is earliest indicator of MI. Then synthesis of heme enzyme ALA synthase condenses glycine with succinyl CoA from delta amino linolenic acid, and it is the key enzyme of heme synthesis. And in the synthesis of purine, also it will give the position of C4, 5 and N7. It will give these places in the purine ring. Then synthesis of glutathione. So synthesis of glutathione is again important thing. Glutathione is a tripeptide formed from glutamic acid, and glycine. These three things are responsible for formation of glutathione. So glycine as a conjugating agent, and as a conjugating agent, it will react with. Let us write the reaction over here. Benzoyl CoA and glycine, they will react to form puric acid. Form puric acid. So through a conjugating reaction, and it will also form glycolate and glyco deoxydiolate. Those all things will be also formed. So in benzoyl CoA and glycine on reacting, they give puric acid. So bile acids, glycine is used to conjugate the bile acids to produce the bile salts, and glycolic acid and glycogenodeoxycholic acid are the main conjugated by bile acids, right? Okay. Then in detail, benzoic acid is used in small amounts as preservative in the food. Glycine is used for detoxification of benzoic acid from puric acid. Then glycine, it reacts with benzoyl CoA to give benzoyl glycine or puric acid and CoA. Conjugation occurs in liver, and so it is an index of liver function. Uric acid. Then glycine as a neurotransmitter. Glycine is seen in the brainstem and spinal cord. Glycine opens chloride specific channels. In moderate levels, glycine inhibits the neuronal traffic, but at high levels it causes overexcitation. The things that are associated is overexcitation. Glycine as a constituent of protein. So glycine is seen where the polypeptide chain bends or turns, and in collagen every third amino acid is glycine. So how much amount of glycine is present in your collagen? Collagen, the main constituent of collagen is glycine. That is, can say approximately 30% of the amino acids of collagen is glycine. 30% is glycine. So glycine is responsible for all kinds of bends and loops because it is such a small structure, so that your molecule can take a turn at particular place. So for bends and loops, glycine is needed. Then what are the metabolic errors that are related to glycine metabolism? First of all is Ketotic hyperglycemia is due to the defect in glycine cleavage system. Glycine level will increase in blood, urine, and cerebrospinal fluid. Severe mental retardation and seizures can be seen. So, this is first type, and there is no effective management now. Then primary hyperoxaluria, increased excretion of oxalates is observed, that is approximately 600 milligram per day or compared to a normal 50 milligram per day. The oxaluria is used to increase production of oxalates, and it's an autosomal recessive trait. The disease is because of protein targeting defect. So, protein targeting effect in protein synthesis, the glycine, when it is in the process, then in the process of transaminase reaction, it will be defective, and that transaminase reaction will further lead to protein targeting defects, and it will lead to primary hyperoxaluria, right? So it is because of the defect in transamination reactions. This leads to the increased pool size of glyoxylate and excess production of oxalate, renal deposition of oxalates with nephrolithiasis, renal colic, and hematuria. External oxalosis may be seen in heart, blood, and bone. In type 2, primary hyperoxaluria is a milder condition causing urolithiasis and results from deficient activity of cytoplasmic glyoxylate reductase. Then there should be a management. So primary management is to increase the oxalate excretion by increased water intake. So try to minimize the dietary intake of oxalate containing things, that is vegetables, sesame seeds, tea, chocolates, beetroot, spinach, and rhubarb. In normal person, oxalate can rise from, in normal person also it can rise, glyoxylate metabolism from ingestion of leafy vegetables, from ascorbic acid degradation. Third source is minimal in human beings, like from ascorbic acid, that is quite minimal. And this is it for today's class, and we have learned about tryptophan and glycine metabolism quite in detail, and hope you have understood the things quite clearly, how the tryptophan is metabolized and how the glycine is metabolized. This we have seen clearly in today's class. Hope you have enjoyed. If you are having any doubt or any query regarding the topic, you can very well write in the comment box, and I'll be going through the comment. And if you want to the particular topic in biochemistry, mention those topics also, and we'll be trying to cover those topics. And lastly, if you like my video or you gain something, so please give me a thumbs up, and please subscribe our channel, that is Let's Crack NEET PG, and don't forget to hit the bell icon so that you will be getting all of the notifications for the upcoming video. Lastly, please don't forget to use my referral code, that is FB01, and by using my referral code FB01, you will be eligible to get an additional 10% discount in your plus subscription. So that is it for today's class. Hope everyone enjoyed, and that is it for now. We'll be meeting again in the next lecture, and we'll be discussing some another interesting topic. Bye.